Alright, so this is our smooth muscle video. So before I begin on smooth muscle, I just wanted to mention one quick thing, and sorry if I'm talking fast, I'm trying to cut down the time on these videos so that I can make it more condensed. Is that I mentioned in the T-tubule we have the triad here with the sarcoplasmic reticulums on both sides. I apologize, and my video quality has gone down because I'm kind of like pressed for time. I'm trying to fit in some studying of my own and stuff. So I apologize for leaving something out. Really kind of is a bummer that I did that. I should pay attention better. But the T-tubules, when they carry down the depolarization of the cell through the sodium channels to reach the inner parts of the cell, they're going to activate a receptor down here at the end of the T-tubule that's very near the sarcoplasmic reticulum called the DHP receptors, dihydropyrimidine receptors. These receptors are activated by voltage, so depolarization. They're going to let in calcium from the outside of the cell, okay? Or inside the T-tubules, whichever you prefer. The calcium from outside the cell is not enough to contract the skeletal muscle fully. So I'm talking about skeletal muscle here again. But if you let in just enough, you can have some receptors on the sarcoplasmic reticulum called the rhyodine receptors. Rhyodine receptors. I forget how it's spelled. That's going to open up from the calcium that is brought in. It's going to stimulate it to open, and it's going to let calcium out of the sarcoplasmic reticulum, which there is a lot of calcium in the sarcoplasmic reticulum in smooth muscle cells, and that will be able and allow you to contract all of your skeletal muscle fibers, but not enough with just the one that comes from the DHP receptors. So that's that. Alright, so smooth muscle, getting on with it. Smooth muscle operates, I'm going to go from, kind of going to do something different. I'm going to go from start to end, and we're going to pick up on the mechanism of how smooth muscle contracts kind of towards the end of the video. So if you were just looking for that, you can skip to the end, but from the start we're going to talk about how the cell is activated to contract, or how it's inhibited. Okay, so our first mechanism of stimulation for muscle cell contraction is our stretch-induced calcium release. Okay, so our stretch-induced calcium release means that there's going to be a receptor on the outside of a smooth muscle cell called the calcium receptor. I believe it's an L-type receptor, but it has a special feature to it, or not a receptor, a channel, but it has a special feature to it which it can sense that a smooth muscle cell is expanding outside of what it normally would be able to. So the cell is becoming too big, and what's it going to do to counteract that? It's going to let in a bunch of calcium from extracellular space. And what's that going to do? It's just going to be like in skeletal muscle. We're going to see from our mechanism, once we get to that, is that when calcium is released inside of a smooth muscle cell, we're going to get contraction of the smooth muscle, okay? So this makes sense because unlike our skeletal muscles, which just contract and shorten in one direction, like this with a bicep curl, our smooth muscle cells are going to contract in a circular, kind of twisty motion. So when they get stretched out too big, they're going to need to clamp back down. Okay, so that's that. I just realized I say okay a lot in these videos. Kind of hate myself for that right now. Alright, so our second mechanism is going to be, it should be familiar to us, and this may create kind of an epiphany for some of you guys with why we memorized and learned all those mechanisms of the autonomic nervous system receptor classes. Okay, so we got our second mechanism being a receptor binds a chemical messenger, a neurotransmitter, or a free-floating chemical messenger like epinephrine, and it activates a second messenger, G protein, called GQ. GQ might sound familiar. I said Gentleman's Quarterly for the type 1 mechanism in our autonomics video. It activates a couple different things, but it's going to increase this level of IP3. Okay, so I didn't mention that as much earlier in my lecture on autonomics, but IP3 is increased by this GQ protein. And thus, the IP3, through a couple different mechanisms that we don't need to know, increases calcium concentration in the cell. So it's going to let in calcium actually from the sarcoplasmic reticulum. Why did I put 2 plus there? Just sarcoplasmic reticulum. Okay? And that is going to induce the formation of something called calmodulin, which we'll get to here in a second. I'm just going to go to type 3 and then we'll talk about the way they work. Okay? It's also going to increase the activity of protein kinase C. And protein kinase C inhibits or downregulates the activity of myosin light chain phosphatase, which I will also get to here. Our type 2 or type 3 mechanism, where GI or GS, which you may remember from the type 1 and type 2 mechanisms respectively, inhibits or upregulates the level of cyclic AMP in the cell. And thus, it's going to increase or decrease the amount of protein kinase A in the cell, which will inhibit the myosin light chain kinase. Okay? So, how does this all work? Sounds fancy, right? A lot of big words, a lot of acronyms. What happens in the smooth muscle cell to make it contract? So one of these things is going to lead, if we're dealing with a contraction, one of these things is going to lead to calcium being let into the cell from either the outer cellular matrix or from the sarcoplasmic reticulum. Okay? And with that calcium, it's going to be free floating in the cytoplasm of the cell. And when I said that in skeletal muscle, calcium bound to troponin to pull tropomyosin off of our actin binding sites, our smooth muscle cells are going to be similar to that, but they're not going to have troponin and tropomyosin. They're going to have something else, and that something else is calmodulin. Calmodulin is going to expose our actin binding sites in smooth muscle. So through one of these mechanisms, pick your one here, you're going to eventually lead to an increase in calcium if you're going to contract, and calcium is going to activate calmodulin. Calmodulin is a protein that can go ahead and activate an enzyme called myosin light chain kinase. So what's a kinase do? A kinase puts a phosphorus group on something, right? So in skeletal muscle, our rate limiting step was, okay, do we have enough calcium to go ahead and uncover our binding sites from actin? In this, it's going to be similar. It's going to be enough, do we have enough calcium, but it's also going to be, do we have the calmodulin to go ahead and put this phosphate group, or to activate the myosin light chain kinase, to put a phosphate group on the myosin head, which is going to allow it to bind to actin. So this is our myosin fiber right here. And they're arranged a little bit differently in smooth muscle cells because it's not like in skeletal muscle where we're just contracting one way. In smooth muscle, we're trying to contract, twisting and narrowing things down. So this is some of our buzzwords. This is a dense body. It's in the cell, smooth muscle cell. They can be attached to the cell membrane or just free floating in the cytoplasm, but all these dense bodies are going to be attach to each other through the thin filaments. And that's important because they pull against each other to, or pull together against each other to actually make the cell clamp down and contract. So right here we've got our dense body, and our dense body is kind of analogous to our Z line in our sarcomere. Off of it comes our actin, just like in the Z line in the sarcomere. And floating out in the cytoplasm is our dense, or our heavy chain of myosin. And on myosin, we've got our myosin heads. Okay, those are supposed to be heads. And there's lots of actin proteins coming off of this. Now, the myosin head can't bind to the actin binding site in this case unless it is phosphorylated by myosin light chain kinase. So unless we have calmodulin allowing myosin light chain kinase to phosphorylate the heads of the myosin thick filaments, we are not going to be able to make a cross bridge to our actin site, our actin binding site. But when we do, we can go ahead and proceed by the same mechanism that skeletal muscle contracts by. So we can bind, or we have an ATP bound, it can dissociate and release that phosphorus group and ADP, and thus pull the, I guess I got that kind of heads going the wrong way. It's going to pull that way, and that way. It's going to initiate a power stroke when that ATP cleaves off and pull the thin filaments towards whatever way you're wanting to go. Okay? So, an ATP can come in again if this chain is still phosphorylated by myosin light chain kinase, and an ATP can unbind it. When an ATP binds, just like on skeletal muscle, it's going to detach, and it's going to find a new home if it's still phosphorylated on another actin binding site and keep crawling up the chain, because it's going to attach to a new binding site. That ADP and phosphorus is going to cleave, giving it energy to do the power stroke, and then it's going to dissociate, and a new ATP can come in and bind. Okay? If we do not have that medium light or myosin light chain kinase there, we're not going to get this phosphorylation. Okay? So the state in which we are not having it, we cannot bind the actin binding sites, and we get no smooth muscle contraction. So we have to have a way to relax smooth muscle, just like we have to have a way to contract it. So the way that's done is using a different enzyme called myosin light chain phosphatase. So a phosphatase is something that cleaves a phosphorus off of something, or a phosphate, excuse me, off of something. So this enzyme is going to take this phosphate group off of the myosin head and basically not allow it to form a cross bridge, thus not contracting the cell. So we've got the way we regulate this is through the use of the second messenger system and calcium. Okay? So in our type 1, I said that an increase in IP3 was going to lead into an increase in calcium, which is going to form calmodulin. So IP3 is going to go ahead and increase, and it's going to induce a receptor on the sarcoplasmic reticulum in the smooth muscle cell to go ahead and open up and release its calcium. Alright, so simple enough. And when that calcium comes out, now we've got the ability to form the calcium calmodulin complex that goes ahead and activates the myosin light chain kinase protein. And like I put a little kind of note here, is that the medium or the myosin light chain kinase is pretty much troponin. It's analogous to troponin in skeletal muscles because it allows the actin binding sites to be uncovered. Okay? So we also have something else going on here. So this is the type 1 GQ protein receptor mechanism. Okay? And where was this used at, or what kind of receptors was this used at in the autonomic nervous system? Try our alpha-1, M1, M3. Ring any bells? Because M3 makes you pee, which should basically become my slogan by now. This M3, I said, was going to go ahead and induce smooth muscle contraction. And it did that by increasing IP3 and thus increasing calcium, which allowed calmodulin to activate the myosin light chain kinase. Conversely, we've got something that also helps us block the opposite way, or block the inverse of this pattern on this. So we also said that we increased protein kinase C in this receptor type. Well, when we do that, protein kinase C is going to, when it's activated, it's going to inhibit myosin light chain phosphatase. So if you inhibit the thing that makes it relax, you're only going to be able to go one way. You're only going to be able to contract. So this receptor type is very good at making smooth muscle contract. Okay. Now we've got the type 3, and I said I'd come back to it. We've got our GI and GS receptor mediated response. We are going to either increase or decrease the amount of cyclic AMP, which is going to increase or decrease the amount of PKA, protein kinase A, which inhibits myosin light chain kinase. Okay? So our type 1 and type 2 both are going to work here. But they're going to have different kind of effects. Okay, so what was type 2? Type 2 was our beta 1, beta 2. And then type 3 was our M2 and our alpha 2. Okay. So these aren't used as much, the type 2 isn't used as much, but you can see that we're going to go ahead and be able to regulate our phosphorylation of this myosin light chain kinase and be able to go ahead and inhibit or induce smooth muscle contraction. So which one does what? Well, it should be self-explanatory from the receptor types and what I said. So I said M2, it brings things down, right? It's inhibitory. So this is going to decrease the amount of protein kinase A, and the protein kinase A is responsible for either inhibiting or inducing or allowing the myosin light chain kinase to phosphorylate the myosin heads. Alright, so that ends my spiel on that. The other thing we didn't talk about yet was there's another way to induce smooth muscle contraction. So I said that protein kinase C can inhibit myosin light chain kinase phosphatase, so that can go ahead and increase your amount of contraction because it's inhibiting this, the thing that lets us relax. How are we going to be able to, what if we just want to relax it? Okay? And that's a really good answer, or a really good question. And the way we get around this is we have a chemical messenger called nitric oxide, NO.